region surrounding a city in which there are at least 2,500 persons. Rural areas are the opposite, as they are defined as areas having less than 2,500 persons. These regions are defined by low population density and large amounts of undeveloped land. Although these locations are quite different, the ecological community or group of interacting or potentially interacting species demonstrate the overall connection between initial species analyzed as well as additional organisms that share the same habitat. The species that was initially chosen for this analyzation were mushrooms, specifically Pluteus cervinus or deer mushrooms. This fungi species was specifically chosen as in particular, these mushrooms are known to be greatly dispersed throughout the United States. And although they are greatly dispersed, this study solely focused on their abundance or lack thereof in specific rural and urban communities within the Washington, D.C. metropolitan area, specifically Washington, D.C. and Prince George's County or PG County, Maryland. At first, the concentration of this experiment was going to be the measurement of biodiversity within the two different study areas. However, the proposed research question became measuring the differences in the two selected communities. If deer mushrooms are located within a rural wooded area, then their population will be significantly greater than the total found in an urban area. That was the initial hypothesis. Although mushrooms were initially meant to be the main focus, a candidate species measured in both study areas as well were ants, specifically Memorium minium, which are little black ants. This secondary species was then chosen to be the primary focus due to their abundance in both areas. An additional species found that was not analyzed, however, was noted was the Colicidae, which are mosquitoes. This was not a surprising find, as they are also dispersed throughout the United States. And research states the warmer temperatures make them pass through their life cycle faster. So more are laying eggs and more eggs are hatching. Therefore, that's why you're finding an abundance of them. This hypothesis may have seemed broad initially, however, there were variables utilized that were part of the overall method, like any other experiment. Science Notes specifies the differences between independent and dependent variables, in which independent variables are the factor the researcher changes or controls in an experiment, and dependent variables are the factor that changes in response to the independent variable. This study included these elements as well as controlled variables, all factors that helped verify the accuracy of the initial hypothesis and measured the differences in species richness and evenness within the two populations. Concepts of biology defines biodiversity as the variety of a biological system, typically conceived as a number of species, but also applying to genes, biochemistry, and ecosystems. Biodiversity is in relation to humans as well as animals. According to the Center for Biodiversity and Conservation, biodiversity includes humans and human cultural diversity. However, use the term biocultural to describe the dynamic, continually evolving, and interconnected nature of people and place. This study is specifically focused on insects, however. Humans and their increasingly detrimental physical role in the environment impact these communities greatly, which will be discussed in the results. As stated, statistical data is imperative during experiments, specifically when analyzing biodiversity measures. The three chosen biodiversity metrics included species richness, S, Shannon index, H, and evenness, E. S was based on the actual number of species within the chosen area. E was based on the relative numbers of individuals per species, and H referred to the richness and evenness of both species. Raw data tracked during the sampling is listed in the table that is being presented. Systematic random sampling was utilized with this experiment in which both study areas were tracked. This methodology is defined as a way to reduce the amount of effort necessary to navigate among sample units, in which there is a random start point. This was thought to be a good starting point to track the differences in these areas. This specific sampling was conducted by transecting, in which the researcher, myself, walked around in both environments for a period of time on the same day, different times, to determine if the species were in each community or missing or inferior in one location. Due to the simplicity initially chosen for this particular experiment, the only tools used as part of the initial methodology regarding the mushrooms were a measuring tape, only to be used to identify the distance between each mushroom when found. The fungi, being a large species, were simple to count. However, the total number of ants within each plot or sample area transected were scientifically estimated, as they were too small and constantly moving to accurately provide an exact number. Within the urban area, plot 1 had approximately 40 ants, plot 2 had approximately 35, and plot 3 had around 20. Again, this is for the urban area. Sample size totaling 95. Plot 1 within the rural area, however, had 60 ants. Plot 2 had around 75, and plot 3 had around 80 ants. Sampling size totaling 215 approximately. The rural area held the greater mean, totaling approximately 72 ants, where the urban area held the greater variability. Various factors were analyzed in addition to the sample size just described, including the sample mean or average population number in each area for the species, the sample variation, the sample variance, the standard deviation, the standard error, as well as the confidence intervals for the low and upper limits of 95%, as indicated in tables 2 and 3 as presented. As clarified by the data, the confidence intervals did not overlap, as the means, the lower limits and upper limits within the two areas were significantly different. Environmental factors that may account for the differences was the actual environment itself. The urban or city areas have greatly been impacted by deforestation and a significant increase of housing and commercial building infrastructures, as we all know, human activity. According to Swartnorth, overpopulation has resulted in the increased exploitation of resources to provide food and water to the growing population, and thus, in turn, has resulted in soil degradation and species endangerment. That was from Maker in 2016 research. This could explain why there was a decrease in the number of ants in the chosen plot area within the urban community, that within the royal community, the difference. In addition, the deer mushrooms were only present in the royal community, most likely due to the abundance of grassland, in which their spores were able to grow significantly because of the damp soil and lack of human population and impact. Lands within the urban area are constantly being removed or deforested, leaving no room for species to live, reproduce, or just simply grow. Additional factors may have included the variations in air temperature, humidity, wind speed, and direction, as well as the amount of precipitation within the two population areas. According to the U.S. Environmental Protection Agency, also known as EPA, hard, dry surfaces in urban areas such as roofs, sidewalks, roads, buildings, and parking lots provide less shade and moisture than natural landscapes and therefore contribute to higher temperatures. Although you may ask how this actually relates to the species themselves, there's always a connection. Research has proven that urban streams are hotter on average than streams in forested areas just as an example, which can directly affect the quality of the soil within these areas, which ultimately impacts the growth of the fungi species, such as the mushrooms, or possibly the number of insects present within these areas. The biodiversity metrics results within this study may not provide all the answers, and some variables may be adjusted if this experiment was to be repeated, such as the independent variables and some of the controlled variables, such as the number of plots utilized and the methods exactly chosen. However, this experiment, if anything, provided a beginning point for additional studies that will allow other ecology researchers to identify more factors and thus answers to continue proof in favor of this initial hypothesis.